let's move ahead now to Dr. George Roderick, who is the William Maurice Hoskins Professor and Chair of the Department of Environmental Science, Policy and Management at UC Berkeley. His research focuses on biological invasions and the history and structure of populations, both especially related to biodiversity science and global change, which is right up the alley as far as this committee is concerned. He addresses both basic and applied questions and takes advantage of opportunities provided by especially the Pacific Rim, Pacific Islands, and California. So with that, let me turn it over to Dr. Roderick. It's all yours.
Okay, thank you. Can you hear me fine?
Yes.
We're good. Okay, so I will give just some examples, largely taking off from where Dr. Lodge left us. And, you know, I have to second everything he said. What we know from islands is that invasive species, introduced species are common. For most of these species, we don't know the impacts, especially non-target effects. Islands and communities are all connected. The social economic networks, in addition to what we know about biology, are really fundamental, and there are many opportunities for studying these things on islands, and I'll come back to those points at the end. Okay, here we go. Many remote islands gain their species through dispersal, of course, from elsewhere on Earth, but they also, if they're remote enough, can accumulate species through evolutionary processes, and sometimes this can happen relatively quickly. Here's just a figure that shows on the x-axis distance to a remote island and how connected they are. If there's a lot of gene flow or migration to these islands, such as invasive species that are coming and going with humans, little divergence develops. But if the populations are isolated over time, divergence can develop. And I think there's some potential for rapid evolution, and we'll see that especially with symbionts and Wolbachia in a minute. The islands that I want to talk about are these, and they're remote islands in the Pacific. Hawaii, the Society Islands, especially Tahiti, and just above Tahiti, you don't see it on the map, but an island called Moorea and one called Tetiaroa. And these islands have a lot of advantages for studying introductions and removals and their consequences, largely because at least some of the ecological communities are simple, but also they're easy to monitor. They're, circumscribed, and we have people on these islands who are watching for ecological and economic impacts. Just to give you an idea of what we're talking about, this is a survey of numbers of Hawaiian species, total endemic, which are those that are only found in the Hawaiian Islands, and then non-indigenous, that would be things that come from somewhere else. And just to look at the bottom line there, for insects and spiders, for example, there might be 8,000 species in all of Hawaii. 5,000 of those are endemic, which is actually quite a high number. And then about a third of the species in Hawaii, for example, are non-indigenous or introduced or invasive. And this number of about a third also happens to apply in California, where about a third of our arthropods, terrestrial arthropods, are introduced. So these are large numbers, and as Dr. Lodge said earlier, invasive species are a big component of what we deal with in ecological communities today. Also, as was pointed out earlier, it's not just these biological communities in isolation, shown on the left, natural ecosystems, but they're also modified ecosystems, and all communities on Earth, of course, are somewhat modified. And as was pointed out earlier, the networks of transportation and connectivity of people, of course, are moving things around in addition to the natural dispersal. In fact, have really augmented the homogenization of the biota on Earth. So just to give you a few examples, this is list of plant species on the island of Moorea that was collected as part of the Moorea Biocode project, which was a Gordon and Betty Moore funded effort to catalog all the taxa and get DNA from all of them on this one island. And these are just plants. On the top you see the native species in red, those that are just found in the Society Islands, and then the alien species per sampling effort on the top. And you can see that, you know, there are a lot more alien species than native species. On the bottom, what's interesting is we can see that a lot of those alien species are cultivated. That is, these aren't just weeds shown in the blue dashed line, but they're actual natural, they're actually purposeful introductions, cultivated species. So in addition to, you know, the invasive things that we worry about that come in on their own and cause damage, much of the biota and flora and fauna of these islands is actually purposefully introduced. Here's another example. This is work by Laurie Henneman on Kauai. And what she did is to rear thousands of caterpillars that she found in the forest, 54 endemic species and 4 alien species. She let these live in the laboratory until either they emerged as moths or a parasitoid popped out. And what she found was that a great proportion of these actually were parasitized. And 83% of the parasites that came out were organisms that had been introduced into Hawaii previously for biological control. And there were also some accidentally introduced species. So as was pointed out earlier, these non-target effects can be very important. These are organisms, these parasitoids were introduced to control largely pests of sugarcane and other crops, but they've made it into the endemic fauna as well. And from these sorts of data, food webs were produced, and you can see the effect here of introduced organisms. Those are in teal, the accidentally invasive or the species that got there accidentally in orange and the native ones in black. Well, it's possible to remove species from islands, and this is an example of the glassy-winged sharpshooter that was a major, or is a major pest here of citrus and grapes in California. It's bad in California because it transmits this bacterium, Pierce's disease. And here are just some maps for its distribution in California. It has a relatively short life cycle. What's important in this story is that the eggs are laid on the leaves of the plant. In about 1989, this species came to California, and then in 1999, probably in association with the importation of certain citrus plants, it made it to French Polynesia, these islands in the South Pacific. And I'm going to show you a little video here of the impact or the numbers of this glassy-winged sharpshooter. So what you see in this video coming up is not rain, but it's actually the honeydew, or what's coming out of the back end of these plant hoppers as they suck sap. So here's a sunny day at the port in Papeete, French Polynesia, and you can see these people looking up in disbelief, not realizing that this is actually the poop that's coming out of the back end of these leafhoppers. Anyway, great numbers. As part of this project, we sampled not only these leafhoppers in French Polynesia and elsewhere in the Pacific, but also elsewhere, and there was great concern that these leafhoppers would get on an airplane through baggage or other means and disperse elsewhere. In fact, we found them loaded on planes. Airports, of course, are great insect collecting vehicles because the lights are on all night, the insects go to the lights and drop on the planes and then are shipped all over the earth. I was part of a big project of biological control, and this is an example of how we might use these eradication efforts or control efforts to understand something about what would happen if we got rid of species altogether. 2 parasitoids were introduced, and here are just the numbers of nymphs of these parasitoids after different years that they were introduced in these different islands of French Polynesia. Before the study started, the French Polynesian government was quite concerned about non-target effects, and so we had to survey the native leafhoppers and to determine that the parasitoids were of a different size, in fact, and would not harm these native leafhoppers, and that's turned out to be the case. As part of the survey, we found 20 new species in the Society Islands that were not previously known. Here's just a diagram showing the introduction of this parasitoids and the numbers of the glassy-winged sharpshooters. So the numbers here on the upper left, the large red circles are the abundance of the glassy-winged sharpshooter, and then after introduction you can see in August and then September and then the following January the numbers decreased substantially. As part of this project, guided largely by work in the marine intertidal zone where researchers have developed appropriate sampling strategies for these before-after paired comparison type studies, we set up a number of plots that we followed before and after the release so we could actually compare control sites and release sites before and after. And here are some of the numbers. The release sites are the closed circles and the control sites the open triangles. And what's interesting, if you look at the density of hosts on the right there, that in the release sites we got rid of the glassy-winged sharpshooters quite quickly, and the control sites retained glassy-winged sharpshooters for a while. But then, within a couple months, even the control sites, the glassy-winged sharpshooters, were reduced in numbers. So this illustrates that, even if you don't intend to move these organisms around, they do move around. And one way we were able to document that was to look at the spread of this parasitoid, which was not introduced to other islands in the Pacific, but to examine its spread. And so, over the next year, this parasitoid made it to all the islands of French Polynesia, including Easter Island and elsewhere. And this, of course, is not because of the scientists introducing this organism, but because people were moving plants around, and the plants had the eggs of the glassy-winged sharpshooter, and the parasitoids moved with these eggs. So this biosecurity issue is certainly an important one, and studies like this show that organisms get around. In fact, they get around quite quickly, unintentionally. This kind of dispersal is, I think, very important, and I think islands provide an opportunity to look at the importance of connectivity or dispersal. And of course, it depends on the biology of the host and also, or the organism, and also the extent to which people are moving those organisms around. Here's an example of Wolbachia frequencies shown in the pie diagrams. On the right, in a butterfly, Hypoluminous bolina, throughout the Pacific. And what you see in those pie diagrams is actually the Wolbachia frequencies can be quite different even among islands that are very close to each other. So this illustrates that this species, in fact, was not moving around much. And so it is possible to get a handle on dispersal, but also we have to realize that different species will differ in the extent to which they might disperse. Okay, islands offer a lot of opportunities. This was a preliminary study done of the atoll Tetiaroa, which is just north of Moorea here, which is just beside Tahiti. So here's Tetiaroa in this diagram. And what James Russell did was to look at Rattus excellens, the Polynesian rat that was introduced with Polynesians maybe 1,000 or 1,500 years ago, and then also these two other rats that were introduced with the Europeans, Rattus rattus and Norvegicus. And he did some some removal studies on these different islands and found that, in fact, it is possible to remove these rats. For islands that were closely connected to each other, the rats could move between islands, but even for these islands that were not separated by very much water, the rats did not move. And again, this is consistent with rats being moved around largely by by humans and their boats. But what these islands offer is the opportunity to, do a species removal and then do resampling afterwards and document the ecological effects. On this set of islands, there's an ongoing study that you probably are familiar with, a sterile insect technique study, again using different strains of Wolbachia and incompatibilities. And this is headed up by Hervé Bosson at ILM, based in Tahiti, working also with researchers at Oxford. And I heard him give a talk on this last week in Moorea, and the results are very promising. And because of the sort of the nature of these small islands shown here above, it looks like it'll be quite possible to, reduce in numbers this species, Aedes polynesiensis. Now, on this island also there's Aedes aegypti, which this project is not dealing with, but aegypti on this island is found more around dwellings. Okay, and the last project I want to talk to you about is a large NSF Dimensions project that's led by Rosemary Gillespie here at Berkeley. And what this project attempts to do is to look at how communities assemble over time using this, what they call the chrono sequence of the Hawaiian Islands. And on the right there's Kilauea, the youngest of the active volcanoes. And as you move northwest in the Hawaiian chain, the volcanoes get older, the islands get older. And so this represents presents a series of ecological communities that vary in age. And so one can ask, well, what's the importance of dispersal or connectivity compared to, say, adding species by evolutionary processes over this structure? And this is just a diagram here showing the idea, which is to sample these different age volcanoes, get molecular data from these as well as ecological data, and to make sense of this over time. And I think what's interesting for the Gene Drive projects is that this is a way to sample communities in a comprehensive way that where one might be able to tease apart the effects of individual species. I'll give you some examples. Here's just, for example, earlier work by Dan Gruner, who's at the University of Maryland, showing the numbers of species on these different aged communities. So on the left is younger volcanoes, and then as you move to the right, older volcanoes. And for many species, you get more species in the middle, or more diversity in the middle, that is the middle-aged islands. And this is consistent with earlier work that Peter Vitousek and others have done having to do with the nutrients available in these communities. Anyway, one can sample biological diversity across these islands. This is an early attempt from this island chronosequence to get a handle on connectivity. This is kind of a complicated graph, but the point is that FST is shown on the left, which is a measure of how distinct populations are. The bluish kinds of lines and dots are sap-feeding insects. The orangey kind of lines are spiders and predators. And on the younger islands, what you can see anyway is that there is a lot of, there's quite a bit of distinction among these ecological populations in the sap-feeding insects, but not the spiders. And I just illustrate this to make the point that it's possible to get a handle on connectivity, that is gene flow or migration among these different populations or locations or communities. And this is one of the things that Dr. Lodge presented or noted earlier, that dispersal is going to be important, and we can get a handle on that. Also, in these sorts of studies, it's possible to estimate food web parameters, things like nestedness and modularity, which are two common sort of features of food webs. Again, with looking at island age and how complex these ecological communities are. And then finally, there are sort of meta-statistics. One can look at emerging properties of these ecological communities and ask, are they consistent with what we might predict from first principles, and whether these communities are more or less ecologically stable or not. And this is one attempt to do this, a complicated figure, but at the right we have different aged communities shown across from left to right, and these are sort of whether the species distributions in these communities fit what we might expect from null models just built from principles of energy and relative abundance. So this is just to give the flavor that in these somewhat simplified communities, it's possible to dissect out the impacts of different invasive species or introduced species, and to try to get a handle on what the biological community means for the success of these organisms, and also what these organisms will do or how they will affect these communities. So let me just wrap up then. So here are some lessons that I think we've learned from islands. First of all, invasive introduced species are extremely common. The impacts of most of these species are largely unknown. Some have been studied in detail, but I would say we know relatively little about impacts like non-target effects in many of these communities. These biological communities and islands are connected, and this depends, of course, on the natural history and the dispersal ability of each of these species, but we've seen that introduced species can get around these islands very, very quickly, although some species that you would think should move around don't. And again, this depends not only on the biology, but also, point 4 there, how these species are connected to people's livelihoods. And I think, as David pointed out, when we are manipulating these species, we will have an impact in some way on the people that live in these areas. So finally, what are opportunities for islands? First of all, there's a great opportunity for resampling previous efforts. There have been many introductions in the past, biological control introductions, accidental introductions, and I think we have a great opportunity to resample some of these habitats where these introductions have taken place and where they have not, and to use that comparison to understand what's happening. They're a great place to look at the ecological and economic impacts of removing species or exchanging one species for another. Again, they're well suited for doing paired comparisons and studies where you do something on one atoll and not another. We have a great chance now, especially with more genetic data, to estimate connectivity of populations and understand pathways, as David and others have done for marine organisms. And then I think finally, with new community-level approaches, we can get a handle on what's going to happen to species diversity with these introductions, but also, what the role of species diversity is itself, what the role of species diversity is in how successful we will be in eradicating species of interest. Okay, so I'll stop there, and I think we're ready for some questions.